So, hope everyone is well rested after action-packed first day. Today will also be action-packed. Okay, so it's my pleasure to introduce Dr. Gong. Guys, am I not loud enough? So, Pulin grew up in China, of course, but he went to Riken after his degrees, and now he's at Sydney. So, in the Life for Science sessions, you may want to ask him about all these places, right? Because he's been more places than I have been, even. Yeah. So, but today he will tell us about spatial activity patterns in the brain.
Okay, great, thank you. And as Lois said, currently I'm a associate professor at the University of Sydney in School of Physics. I've been working on developing new methods and theories and models for understanding how the brain computes. So the brain could be, the most complex system in the universe. Do you agree, guys? Yes, okay. So when we look around at our universe, so one thing we should notice is that there are a lot of these sort of emergent coherent structures, okay, because of interactions of many macroscopic these sort of interactions. Okay, so here I'll just give you an example. So this is this sort of like massive, sorry. Get this movie right? So this is a massive, this sort of like a coherent patterns of spiral waves or vortices, as you may say, emerging for the Jupiter's great red spot. Okay, so this is really fantastic. And also, if you look at the molecular level, so here we'll see this signaling RHO, GTP signaling protein, or the membrane of just one cell. Okay, one starfish egg cell, you will see this sort of beautiful coherent structure, so spiral waves. Okay. So they are extremely important for organizing some essential functions of cells. So by the end of today's lecture, I hope you'll get an idea about the importance of those patterns, okay? And also the way how we can analyze those patterns from real data, okay? I will go through all kinds of data, and by the end of my lecture, you should get some ideas about these sort of patterns. So here, this is schematic of cortical circuits. Okay, cortical circuits are composed of many excitatory neurons and many inhibitory neurons. Okay, around 20% of them are inhibitory neurons, and 80% of them are excitatory neurons. They interact with each other, forming complex network. Okay, and the complex dynamics can emerge from those networks. Okay, those dynamics are important for brain functions such as perception, cognition, attention, and behavior. So, and one long-standing observation, established observation, is that actually individual neurons, okay, they behave in very irregular and variable way. So here I just have one neuron. Okay, so this is different trials. So this neuron would spark here, a dot there. They would spark here as well. So this is for spontaneous activity around zero time here. So this is stimulus onset time, and the fun rate, of course, would go up. Okay, but when you look at this, you see again, you can see sparks are very variable. Okay, so sparks are very variable. However, at the circuit level, we'll see some sort of emergent, as I said, self-organized phenomena. So one phenomena would oscillations. Okay, so oscillations are ubiquitous in neural circuits, okay, and also in nature as well, right? So oscillations are extremely important. So here, so there is a beautiful review paper by Bazaki and his collaborator. So oscillations in cortical circuits can be roughly divided into different classes. So here, this is a figure about different classes of oscillations, okay, in the red cortex, okay, for human. So oscillation bands are slightly different, okay, but anyways, so you'll see, so here I have different frequencies, this is different classes. So oscillations can be very, very fast. For this case, okay, this is ultra-fast oscillation. So this oscillation is around 200 to 600 hertz, okay? And then if we go down, so this would be gamma oscillations. Gamma oscillations, okay, from 30 to 8 hertz. Gamma oscillations are extremely important in the brain, okay, related to our cognitive functions, attention, and so on. And then here we have theta oscillations here. Theta oscillations are also important, okay? Anyway, all oscillations, they are very important, okay? As you can see, oscillations can span very fast to very slow band. And this is typical figure, original time series of local field potential of theta oscillations. You'll see this sort of like oscillatory component. So this would be spikes. Okay, spikes would be locked to phase of theta oscillations. Okay. So this is a original trace of theta oscillation. This is a filtered theta oscillation in theta band. And this is an example of in human, this is called alpha oscillation, right? So you will see this sort of alpha oscillations. So those oscillations, they are not independent. Actually, in the real brain, those oscillations, they interact with each other, okay? So there would be sort of like a cross-frequency coupling. So here, so this is an example. Here I have slow theta oscillations, okay? So for this slow theta oscillations, you see you have up, down. Of course, we can see amplitude of theta oscillations. And also for oscillations, we'll see phase of oscillations, right? So this is phase, okay, maybe around the minus pi, this is pi, just give you example. So this phase of theta oscillations, based on this schematic, we'll see phase of theta oscillations would modulate amplitude of gamma oscillations, okay? In that sense, there is a cross-frequency coupling, okay? So this is just about the temporal behavior of those oscillations. So today, I'm going to show you that those oscillations, they're also organized in space, forming complex spatial-temporal patterns. So here, another feature about cortical oscillations would be that is sort of like a periodic component, okay? So you often see, when you do this power spectral analysis, you often see some peaks here, right? One peak here, another peak there. Okay, just give you an example. So these peaks would sit on the top of this kind of, if this is log plot or this kind of linear line. Okay, so this behavior indicates that a periodic, this kind of oscillation, okay, or behavior or the activity would be organized as y over f. Okay, y over f, this sort of component. So this would be another hallmark of brain dynamics, y over f. So if you want to develop a theory, okay, your theory should be able to explain, let's say, peaks, okay, oscillatory component, and also y over f, this sort of dynamics simultaneously, okay? So that's something I'm going to talk about in my second lecture. And then, so this is from a human EEG, and when you look at this is from neural pixel data, okay, for across different cortical areas of mice, you'll see this kind of hallmarks I've just mentioned, okay, peaks. So this one, theta oscillations. Here, gamma oscillations, okay, they sit on top of y over f, this line, okay, indicating sort of like complex dynamics, okay, temporal dynamics so far. So this is a typical time and frequency plot about neural activity or neural population activity. So you'll see, so this is different frequency bands, okay, slow oscillation, and this is around theta oscillation, gamma here. So one feature you should notice is that those oscillations, they tend to occur as bursts, okay? So you see, I mean, give you example here for theta oscillations, okay? You see a hot spot here, another spot there, and so on, okay? So oscillations, they are not sustained or persistent oscillations in the brain. They are intermittent, this kind of bursting activity patterns. So these are main features of realistic, yes? Yeah, so this one, so this is related to this one, right? This is log plant. So this is straight line there. So that indicates there is existence of Y/F, this sort of like behavior in real neural population activity. So which is another hallmark of brain dynamics. Okay, often when people analyzing when analyzing like oscillations, they just ignore this Y/F, this sort of dynamics. But as you will see in my second lecture, in theoretical work, right? So this is actually extremely important for understanding complex dynamics of the brain. Okay. All right. So often people just look at synchronization of different oscillations. So this is just give you an example of theta oscillations. Okay, they look at zero length synchronization of those oscillations. It means that they look at whether or not those oscillations would go up or go down simultaneously. Okay, zero length oscillations, and based on this, people proposed many theories, let's say, okay, about how those oscillations can do some sort of function in the brain. Okay, one idea is that synchronization of gamma oscillations could be important for communicating information in the cortex. Okay, as proposed by Pascal Fries, and also, as proposed by Wolf Singer, synchronization of oscillations could be important for bodying different features of external stimuli. You have one feature, another feature, so different features could be encoded by different these kind of oscillatory activities. And synchronization of those oscillations could encode or could bind the information together. Anyway, so my point is that this sort of people published many papers on synchronization of neural oscillations, and also when we zoom out to look at the whole brain, okay, let's say in the field of neural imaging. In the field of neural imaging, also people only look at zero length cross correlations between different sites. Okay, so you can pick up one site here, which is called a seed. Okay, you will see these slow oscillations or fluctuations. I will talk about this shortly by talking about my research results. And then you look at cross correlations between this side and the other sides. Okay, and then you can map this sort of correlated and anti-correlated these kind of structures. Okay, so this is what people often refer to as functional networks. Okay, based on this, people find many different functional networks like default mode networks and attention networks and so on. So this is one of the first papers on this topic. Okay, correlation-based characterization of brain dynamics. So this is very popular in neural imaging field. Okay, zero length cross correlations. So this paper alone has been cited 9,400 times. Okay, up to now. So this indicates that this is very popular dominant methods to look at brain dynamics, human brain dynamics particularly. All right, but thing is that when we look at the real data, as I said, okay, we often see, okay, so this is slow oscillations recorded from Utah array. So Utah array can be used to record local field potentials of a population, neural population activity. So this is Utah array, 4 millimeter by 4 millimeter. So different site, one site, another site, another site. So this would be the original trace of local field potential. So this is for filtered oscillations in delta band, which is around 1 to 5 hertz. You see, so this for this site, okay, different colors for different sites. And then, because as I said, as soon as you have oscillations, you can detect phase of those oscillations, okay? Based on phase, because phases are important, because phase would tell you the timing, okay, the timing of your oscillation, okay, where your oscillation is at that time moment. And then based on phase values, okay, detected about these oscillations, we can get this sort of phase map, okay? It's just like this Utah array, 4 millimeter, 4 millimeter, we can get this sort of phase map. As you can see, okay, clearly, okay, we didn't do any other additional processing, okay? You will see this sort of beautiful, this sort of coherent structures, okay? So you see this sort of rotational structures, okay? That's something I'm going to talk about shortly. So what it indicates that, although individual neurons behave in a very irregular way, at circuit level, we have these emergent coherent structures, okay? So coherent structures or patterns are the main focus of today's talk. So I will first talk about methods we developed for detecting and analyzing spatial-temporal coherent structures or wave patterns. I will then apply these methods to large-scale data, okay, from local field potentials to imaging data to fMRI data. And we made new discoveries about like vortices or spirals in the human brain, okay? So I will talk about that shortly. And also, if I had time, I would also talk about models and we developed for understanding how patterns can emerge from the cortex. And so this is for today's lecture, and also for more advanced lecture, I'll focus on a new mathematical theory we've developed to understand how this complex spatial temporal dynamics can be used to do flexible and efficient computation. And that theory actually can explain many cognitive functions, okay? Believe me, make sure you will be here, right? So in terms of attention and cortical communications, decision making, and also we apply that theory for understanding deep learning, okay? Because in deep learning network, we have similar dynamics, okay? Those dynamics are extremely important for deep learning, right? That's something I'm going to cover for my last lecture, okay? More advanced lecture by involving more advanced mathematical techniques about fractional differential equation, okay? So fractional derivatives or something like that. So that's why a term fractional neural sampling, fractional neural sampling. Yep.
The neurons are like spiking.
Yeah, that's a very good question. That's actually a question the whole field is asking. Okay, what's the relationship between spikes and local field potential? Because I don't think there's general consensus on this question, but the general understanding is that local field potential is reflection of neural population activity, but more about like synaptic inputs to those local neurons. Okay, of course, this would be related to spikes as well. Okay, there are many review papers on this topic. As I said, there is no consensus, okay, on this question, okay? And also how local field potentials would be related to, let's say, slow oscillations of functional magnetic signals, okay? Still this ongoing research topic. But I will talk about that in the later part of my today's lecture. Yes? Yeah.
Electrophysiological recording, and can we extract from fMRI data some understanding about electrophysiological synchronization?
I mean, there are some studies they're trying to look at how these local field potentials are related to fMRI signals, right? I think I will talk about that paper. There's a Nature paper, and they found that particularly gamma oscillations are closely related to slow oscillations of functional fMRI signals, okay? But as I said, okay, those oscillations, they are not isolated in the brain. They have this cross-frequency coupling and interactions. And but exact neurophysiological mechanism how signals recorded by local field potential, right, related to some macroscopic signals such as a F-both signal, right? I mean, that's still unclear. Okay, all right, and and then, I mean, as I said, so this is a scenario in terms of emergent coherent structures from irregular individual neuron activities. Okay, this scenario is very analogous to the emergence of coherent structures in turbulence. Okay, so you can see these beautiful vortices or spirals emerging from turbulent dynamics. Okay, so this is a classical paper on this topic. And just by drawing this link, okay, link from different field, okay, this is the way how we generate new ideas. Just by drawing this link from turbulence, okay, we've developed a new methodological framework to look at emergent coherent structures with patterns in the brain. So particularly, we borrow the idea about critical points in turbulence. So critical points in turbulence, right? I mean, as demonstrated in this paper, critical points are points where streamlines, okay, slopes is indeterminate and the velocity is zero, okay? Because in turbulence, so this is real experiment, this is theoretical methods, okay, to detect those coherent structures, first thing you can do is that you just have a velocity fields, okay? Just like velocity fields. So for this case, so this is a real experimental observation of turbulence, and you will see some sort of these emergent coherent structures. And here, so this is you have velocity fields, okay? Velocity fields, they are vectors, right? You have many little vectors about velocity of and overall you can have a field. And for this field, you will see, so here this is one critical point, this is another critical point. So this critical point is called a focus, okay? This is focus, so this S represents sandal, and this is sandal, this is node, okay? As well, this critical point, we have a stable, this kind of a streamline, okay? Or if you prefer, you may say that would be manifold, okay? And also you have unstable manifolds, okay? And so this is focus point, critical point. So you have this unstable behavior, and that would go to this stable focus point, okay? This focus actually is spiral, okay? A spiral wave. So just by adapting this concept from turbulence, we developed methods, okay? We developed methods for detecting those coherent structures. We have a methods paper published around 6 years ago, okay? If you want, okay, you can try this methods toolbox, okay, by downloading our toolbox and run some sample data associated with that toolbox even today. So the idea is that, as I said, okay, for this time sequence, we have this time sequence of real data, 2D or 3D, okay? X-ray as a function of time. So first, for this data, we do filtering process, okay? Perform filtering analysis, let's say, to focus on specific frequency range, such as theta, gamma, or slow oscillations. And then, for those oscillations, as I said, oscillations, they have an amplitude, and also they have phase, right? And then we use Hilbert transformation, okay, to extract phase of those oscillations. Of course, you can use other methods to extract phase of those oscillations. So here, just Hilbert transformation in our methods, we can extract phase of those oscillations, or amplitude, analytical amplitude of those oscillations. And then, as I said, okay, to adapt some methods from turbulence, so we get come up with a velocity field, okay, in velocity in x direction and the y direction. To calculate the velocity field from the data, okay, and then we adapt optical flow methods in computer science to estimate, to calculate velocity fields of the real neural data, okay? So this is a methods developed in computer science, optical flow methods for detecting velocity fields. And then as soon as we have velocity field, and then we can do very simple calculation. Okay, so first we detect critical points. As I said, around critical points, velocity would be around 0. Okay, and then for those critical points, we calculate Jacobian matrix. Okay, which is 2x2 Jacobian matrix. Okay, very easy stuff. Okay, you are smart guys, so you know how to calculate this easily. And then based on this Jacobian matrix, right? There are some conditions here. Okay, we can test. We can judge. Okay, we can tell whether the critical point is a node, or focus, or sandal. Something I've just mentioned as for turbulence. And then this is a typical unstable node. Okay, this is singularity point. This is unstable node. You'll see activity would flow out from the singularity point. And then we have stable node. For this stable node, activity would flow toward this singularity point. So for this reason, we call this source. Okay, this is source, this is sink. Okay, sink. And then for this one, we have saddle pattern, as you can see. Okay, stable, unstable directions. And also we have spiral wave. Okay, this is spiral out. Okay, it means the spiral wave is unstable, and also we have spiral. So this is focus. Okay, this is a type of spiral. But anyways, so we can now detect all types of wave patterns you can imagine from the real data, right? Of course, in real data, as I said, okay, classically people would detect synchronization of gamma oscillations or theta oscillations, right? Theta oscillations in our framework can be characterized by introducing order parameter, okay? Just like this simple order parameter, because as soon as you have phase, you can introduce order parameter like that, okay? So when this value is close to 0, it means that neural activities, they are desynchronized, okay? When they're close to 1, it means that they are very synchronized. So so this for detecting synchronization, and also we can detect planar traveling wave, okay? This planar traveling wave is a main topic, okay, studied by other groups, such as like Terence Lomsey's group and Sorg Institute. So for this one, we can introduce average normalized velocity. So this is average normalized velocity. Where this order parameter is close to 1, it indicates that all velocity vectors would be aligned in one direction. You have perfect planar traveling wave. Yes. Yeah. I mean, you can interpret this sort of rotational. I mean, because limit cycles would correspond to oscillations, right? So because of those oscillations, we can detect amplitude and also phase, and then we can use this framework for detecting patterns beyond oscillations. Okay, limited cycle is one level below this framework. Okay, based on those oscillations. But in the brain, as I said, those oscillations, they are not limit cycles. Okay, because of this kind of intermittent, this kind of bursting features. And then based on this, we can, when it's close to 1, we have perfect traveling plane wave. When it's close to 0, it means that there's no traveling wave there. All right, we apply this to, as I said, okay, to this Utah array data. This is empty area for marmoset. Marmoset is small, very cute monkey, okay? And so this is the empty area. And because this the animal was anesthetized, that's why we can see this sort of slow oscillations. Okay, this is time, this is frequency, slow oscillations in third time range. As I've just mentioned, based on this, we can detect all kinds, this kind of the trace, okay, of local field potential, and then we can extract the amplitude and the phase, okay, as I just said, we'll have a phase map here. Based on phase map, we can then apply our turbulence motivated method, okay, to detect velocity field as such. And then based on this, okay, this is real data, so we found all kinds of complex wave patterns from the real brain. Okay, so that's really cool, I think. So this is a source pattern, as I just said. Okay, this is a critical point. This is sync pattern. Activity would converge to that singularity point. And then we have spiral pattern and such. Okay, so you have this sort of spiral structure for organizing activity in that local area. And then we have sandal pattern as shown there. Okay, from local field potential recordings in the empty area of marmoset. And also, I'm been collaborating with Chang Sung Zhou from Hong Kong, and also Thomas Knopfel from Imperial College London. So we applied our framework to the whole cortex data of anesthetized mice and also awake mice. So this is based on optogenetic voltage imaging method developed by Thomas Knopfel. Okay, so this is a state-of-art imaging method. So we found, I mean, for anesthetized animals, you will see we have this kind of boring, traveling waves, okay, synchronization, and also this kind of global homogeneous patterns. However, for awake cortex of mice, we'll see more these sort of like complex waves, okay, such as sync, source, and the saddle patterns, as I've just defined. Okay, so this result indicates that the complexity of waves would increase when animal the brain state, okay, change from anesthetized state to awake state. Because conventionally, you just look at also whether your brain would have activity or not, right? And then you can judge whether the brain is in anesthetized state or awake state, or based on like coherence or functional networks, okay? But here we've come up a new set of wave-based indices for judging whether the brain is in which stage, okay, in anesthesia stage or wakeful stage, okay. So I think this is the application of our methods. Yes. Yeah. Oh, this is the left hemisphere, the whole left hemisphere of mice. Oh, yeah, yeah, yeah. Yes, yes, the velocity vector fields, as I just said. This is for slow operations. Yeah. Yeah. Oh, okay. That's a very short period of time, or rather, like sometimes it could be around 200 milliseconds. Sometimes could be like 50 milliseconds or something like that. Very variable. So their durations are very variable. Locations are also very variable. Very variable. I think that's the whole point of the brain dynamics, right? Very variable, very non-stable. But there is some sort of organization principle here. I will talk about it now. Okay. So this, I'm really excited about this. Okay. Actually, so this is, and then we applied our method to look at the fMRI data. Okay, slow oscillations from 0.01 to 0.1 hertz, right? So this is very slow oscillations. As I said, conventionally, people just look at cross correlations. You pick up a siege, and then you look at the cross correlations between different sites, and then you come up with functional networks, and then you link those functional networks to different brain functions. So that's conventional framework. So here, we applied our methods to look, I mean, this is a trace, okay, from one side, another side. You see these sort of slow oscillations or fluctuations, and then you can extract phase, okay, something we've just learned, okay. You see, phase would be, can be used to reliably track the fluctuations of those slow oscillations. So that's important. Make sure that the phase values are indeed the phase values of your oscillations. And then based on this, okay, we get phase map. So this is phase map I just mentioned. So this is 2D flattened cortical surface, okay, from HCP. Yep. Oh, this is fMRI data. Yeah, fMRI data, yeah. Slow oscillations of fMRI data. So we use data from HCP project, right? And so fMRI data, that's why it's, here we have slow oscillations, okay, phase of those slow oscillations. And then we have this 2D, this kind of upfolded cortical surface, okay, structure obtained by HCP. And then based on this, we have phase map, okay, based on phase map. And then we can apply our framework. And then you can see, here you'll see these kind of little arrows or vector fields, okay, based on vector fields, you'll see, so this is one singularity point, okay, singularity point. This is another singularity point. So here, and then this is just the vector fields, and then around the singularity point, so you'll see that the vector fields, for this case, vector fields would rotate, okay, anti-clockwise, in this anti-clockwise manner. So this means that here we have a anti-clockwise spiral, okay, anti-clockwise spiral organized around that phase singularity point. And here we have a clockwise spiral organized around this phase singularity point, okay? So it means that we found spirals, okay, all kinds of many spirals concurrent in the brain. So spirals, as found here in physics, they are called vortices. Okay, they are called vortices. Or in condensed matter physics theory, they are called topological defects. So here it means that we found vortices or spirals in the real brain. And also, so this is another indication that, so this is about phase, right? That this is amplitude for a distance from the singularity point. You'll see amplitude will increase, okay, if you move away from that singularity point. It means that this is a true vertex or spiral as found in the real, in other physical systems. Yes. The velocity is zero. Okay, velocity is zero. And also another feature is that phase is undefined, because that's the place where different phases would converge. Okay, that's why it's singular in that sense. Yeah.
Is it like each arrow on those plots is particular voxel that was recorded? With
Yeah, exactly. Yeah, project on 2D space.
Okay, thank you.
So, and another feature I wanted to point out, you see those spirals, they tend to located around the boundaries between different functional networks. So this is one functional network, this is another functional network. So these dashed lines indicate boundaries between different functional networks. So those spirals, they are located around boundaries. So this feature actually is very important because think about it, as compared with the plane wave. So spirals would have extra degree of freedom of this rotation, right? Because of this rotation, and so because of this strategic or this special location around boundary, and the spiral waves, right, can organize activity to flow out from one area and flow into another area, okay, because of this rotation. So that's why I think spirals are often found around the boundaries between different functional networks. I will talk about that. And also here I have a movie to show you. As you can see, those spirals, they are not static. The centers of those spirals would move, okay, will move across the cortex, okay? So somewhere, sometimes around the front area, and quickly it can move to the sensory cortex. So this is quite interesting. So they rotate around the singularity point. Meanwhile, the center of singularity points would propagate across cortex. So this kind of complex spatial-temporal dynamics. And this rotation, as I said, the rotation, these dynamics are very non-stationary. Sometimes the spirals would rotate very quickly, and sometimes vertex or spirals would rotate very slowly. Okay, these sort of non-stationary features. So this is trajectories, the centers. Okay, the trajectory centers across this 2D cortex. You will see trajectories are very variable. Okay, so in this sort of variable, this kind of wavy path, actually, and then we calculate squared displacement. Because you have trajectory, you can figure out displacement and mean squared of that displacement. And then you can see this is time delay, and you will see this sort of a linear line or log-log plot. So this line indicates that the movement of those spirals would follow super diffusive motion. If the exponent is around 1, that's normal Brownian motion. Okay, it means that, okay, so those guys, they would move okay, in a more complex way than Brownian motion expected. All right, and then we can, for example, I will go through this quickly, and then we can see those spirals, they would interact, and these interactions are very important for organizing large-scale brain dynamics. I tend to think this is as large-scale organization principle for the brain dynamics, okay? So here, as you can see, this is an anti-clockwise spiral, clockwise spiral. They interact. Through this interaction, okay, both of them, they would die, okay, disappear, die. And but the thing is that, as you can see from this figure, before their interaction, we have this traveling wave, okay, going through this area, okay, this very nice traveling wave. That's just because of their interactions. After their interactions, you see the direction of a traveling wave would be changed. Originally from left to right, and here from right to left. So it means that interactions of those spirals can be used to coordinate this sort of brain dynamics. And then, of course, we have, this is a full annihilation, and then we have partial annihilation. It means that one of the spirals would disappear because of the interactions. Another one would survive. And also we observe repulsive interactions. It means that you have one spiral here, another here, and then they have these repulsive interactions. Both of them would survive of these interactions. And then, of course, if you have many of these sort of coherent structures, okay, they can interact in a cascaded way. Let's say here you have two spirals, they interact, and this will go on to interact with another one. Okay, so in this way, they can somehow organize the flow of neural activity or brain activity, yeah, in that area, yes. Oh, but when they have the same sign, so here's the example. I mean, they interact, and most often here we will see a sandal point. Okay, so I will show you actually we can link these two behavior to brain function. Okay, to brain function. So so this is related to this, and we can now based on this. This is a left hemisphere. This is a right hemisphere. So this is different tasks, right? And it's collected by HCP project. So this is story listening. This is math listening task. So they ask subjects to listen story, and after that they ask the subjects to do some tasks. Okay, so as you can see, for story listening task here, I have a spiral. This is an anti-clockwise spiral rotating in anti-clockwise way, and then we have a method listening task. Okay, so the same location around the prefrontal area, and you'll see clockwise. So this is anti-clockwise, clockwise spiral. You see the rotation direction would be reversed. Okay, so it means that the rotational directions of those spirals are task specific, because you can use these rotational directions to read out information about task subjects performed. And another thing is that, so this is for left hemisphere. When you look at the right hemisphere, you'll see, they are symmetrical, okay? Because this is left, this right, okay? You get this, okay? Because that's why this rotational direction would be reversed, okay? Because you got to make sure that there is a symmetrical relationship. It's general, okay? So this, but for this task, so this is particular for this area, okay? But for, I can show you, so this is for listening task, but for answering task, okay? So again, you will see, so this, the vortices or spirals in that area, so this one, for this one, this is close to PCC. This one is close to, I think close to attention network, but I forgot the exact area. But anyway, so that, this is another indication. You can see rotational direction, okay, is task specific. Left, right, okay, you have this symmetrical relationship, yes. What? Oh, it was published in Nature of Human Behavior. Okay, I can show you. This paper, yes. Yeah. You can read it. Long paper, about more than 20 pages. Oh, yes. Yeah, exactly. Yeah, that's very good point. That's because, so here, so that's for just one spiral, but if you look at the many of them, they interact, can organize specific direction. I will talk about that shortly. That's very good question. Okay. Anyway, yes, and then based on this rotational directions and specific locations of those spirals, we can actually decode different tasks. Okay, that's like decoding method also used to do some neural decoding. Here we have the performance. This is based on 5%. So this is the performance of the decoding based on spiral locations and also rotational directions. Yes.
Hi. I think this is super interesting. I'm just trying to understand the intuition more broadly. I'm wondering if you see any, different importance between different brain regions or different brain pathways or any individual differences, and what does that imply about how the brain works and how human cognition works?
Yeah. I think we think about, as I said, as another organization principle of brain dynamics, okay? And we demonstrate this is related to brain function. But this is imaging study, right? Everything is causal, but we have ongoing product. We're trying to show this has some causal contributions to cognitive function. And so based on this, we can decode tasks. And also another thing I wanted to point out, as I said, okay, those spirals, they are located near the boundaries between different functional networks or brain regions. Okay, for the reason I've just mentioned, because of the rotational directions, right? Spirals can organize the flow of activity from one area to another area. Okay, we tend to think this is as coordination mechanism of the brain dynamics. And another thing we noticed that those spirals also tend to occur. Another question, yes. Oh, two questions, yes. Oh, yeah. You go, yeah. Yes. For this one, we have the task have 4 conditions. Okay, that's why the baseline, this decoding performance is 25%. Oh, yeah. And also, we try this for working memory. I mean, you will see this kind of accuracy. Okay, yeah. And another thing, as I say, oh, another question somewhere. Yeah. Yeah. I mean, the results we obtained are for like 100 subjects. Yeah, but yeah, based on HCP data. Yeah. And then we have this, we performed a similar analysis for working memory. We got similar results, okay? And also this phenomenon is very robust, okay? If you visualize your fMRI data, just look at the amplitude, you will see this sort of rotational structure. It's not artifact of something. And also, yeah, for different tasks, one thing I wanted to say, okay, so this is actually related to, if you look at this kind of coherent structures or spirals, and then if you do PCA analysis, you can project these coherent structures on 2D, okay, manifolds. You will see this sort of rotational, this sort of structure. So in neurophysiological recordings, okay, this kind of research, people publish many papers. They found that motor behavior would be related to this rotational manifold, low-dimensional manifold, okay? But in our framework, those low-dimensional rotational manifolds could be the byproduct of these spatiotemporal rotational structures, okay? And also these spirals, okay, here we have a clockwise spiral, anti-clockwise spiral. They interact, and as I said, these interactions could be extremely important for organizing the flow of information or the flow of activity. So here, I'll just give you an example. This is clockwise spiral, anti-clockwise spiral. You see, because of these interactions, activity can be guided through their interaction zone, okay? So guide through this interaction zone. It means that this interaction can be interpreted and sort of gate, okay, to encourage the activity to flow through that interaction zone. And also, if we have clockwise, if we have this spirals with the same rotational direction, you see, I mean, activity would be blocked away from the interaction zone. Okay, so this means that activity is prevented from going through this interaction region. Yes?
Yeah, sorry, can you explain how the vectors are calculated? Is it like a temporal pattern of activity, and what scale this is happening at?
Oh, you mean the vector? I mean, vector is just the vector of velocity fields, as I said, okay? Based on those velocity fields, and then we adapted some methods from physics, and then we can detect those these patterns. And this actually, because this is fMRI signals, right? Very slow. So specific frequency range we focus on is from 0.01 to 0.1 hertz. Okay, but as I said, people found that these slow oscillations could be related to fast gamma local field potential oscillations. Okay, yeah. Yeah, sure. I will talk about a similar result from different groups shortly. Okay, so people found something similar independently. Okay, so that actually is really really cool. So this, yeah. Yeah. Yes. Yeah. Yeah. Yes, and that's why we are here together, right? And so this kind of unexpected leak. So, and then you can apply our method to look at your vector fields and to quantify your patterns in a similar way, like what we've done for our data. Yeah.
You talked about the information goes with the flow.
Yeah.
In the phase plane, and I was wondering how would you explain with the spiral and the singularity? What does it do to the information?
Yeah, that's a very good question. So far, okay, we haven't linked to information yet, but we've linked this to behavior. But the link to behavior is sort of macroscopic level, and then we have ongoing project. I mean, we want to look at whether those streamlines do indicate the flow of information by characterizing information carried by those streamlines or flow lines. Yes. Not stuck. I mean, you can imagine that just like what people studied in physics, right? Like in optical systems, people often use vortices to design new computing devices. So those vortices in optical devices, they are extremely important in terms of mixing information, okay? Because you have information coming from here, you have information coming from there. Because of rotation, information can be mixed together. So that's from different fields, from optics. So optics, this is a very active research field in optics. So people trying to design these sort of new computing devices based on spiral waves, okay? And then you see, as you see, I mean, this is just pairwise interactions, and then we have this many interactions of many spirals, okay, that can be used to organize this large-scale flow of activity. And then, I mean, we look specifically around this auditory cortex, because this is a story listening and story answering auditory task, and by applying this interaction rule I've just mentioned, okay, clockwise, anti-clockwise, and then we come up with this sort of, we did find, okay, first we did decoding, we found this kind of task-specific region, okay, task-specific region, and then we applied that interaction principle to look at how this interaction can be used to organize a flow of activity during this specific task. So this is for math listening task. You see, I have this many 1, 2, 3, 4 spirals working together to organize the flow of activity from anterior to posterior direction in this bottom-up direction. And then during task or stage of the task, and then we have this, for most of those spirals, they would reverse their rotational directions. And then now activity would flow from posterior to posterior to anterior direction, indicating there's a flow of activity from top to bottom, okay, bottom up, top down directions. So this flow of activity can be organized by interacting spirals. And also this is a result which is now under review. So we found that, I mean, as I said, this is for different data sets. We found spirals that tend to be located around boundaries between different areas. But this is for different aging groups. So this is for young subjects. Okay, this is for older subjects. As you can see, even virally, I mean, for younger guys or younger subjects, we have more spirals. But for a little bit older subjects, okay, we have less spirals. Okay, which is consistent with the idea I've just mentioned. Okay, those spirals, they could be important for coordinating communications between different brain areas. Okay, so you guys have more spirals. Okay, that's why you're smarter than me. Okay, so because communications between your brain areas could be faster than communication in my brain. Okay, because you have more spirals. And also we've used a phenomenological model. This is phenomenological model. It is not a realistic model. So we have an ongoing project in my group. We're trying to develop this large-scale whole brain model to explain how those spirals would emerge from the realistic model. And so this is just, as I said, we have oscillations, or each oscillator is represented by a phase oscillator, and there's coupling, neighbor this coupling, okay, based on this interaction phase, and then you see these sort of spiral waves will automatically emerge, okay, something related to the metopic. And then, I mean, these interactions, just like what we found in the real brain, can actually organize the flow of activity. So the mechanism we've suggested and validated in this phenomenological model. And let's say here, if artificially reverse the rotational direction of this spiral, and you will see that, and then after this operation, the overall flow pattern would be reversed as well. Okay, so this suggests that the interaction If we look at our data, we'll see this sort of consistent trajectory. Okay, so it's really amazing, okay? And based on that, we introduce consistent path. And then we link that consistent path to memory performance. Because speed oscillations, okay, are believed to be related to memory consolidation. Okay, that's speed oscillations. And we found that these consistent trajectories of those spindle spirals, okay, could be important for memory consolidation. Okay, that's why we need that sort of consistency. And also spirals, okay, for slip spindles are very big, as you can see. Yeah, and as you can see, as you can see, okay, almost it covers around the whole brain. Okay, very big one. Okay, but this only happens, these big spiral waves, they only happen during sleep. Okay, that's understandable, right? Because during sleep, I mean, you don't do anything else, right? You only sleep. And also it had, yeah.
The plot you have on the next slide, I think. What is the score on the y-axis?
Oh, doesn't it? Yeah, this one. So this is a record score because I mean my collaborators they recorded like performance for memory task for subjects, right? I mean you have this memory task and based on the report performance, and then you can come up with a report score, which is a behavior index for that specific memory-related task.
Okay, and for the long-term consistency?
Long-term consistency, I mean, as I said, okay, for slip spindles, the trajectories of those spirals are very consistent, always around the same trajectory. So first time I saw that, I was really shocked. I couldn't even sleep that night. Okay, because it's so consistent, right? I mean, we know that the brain is very noisy, right? But during sleep, we have this emergent, consistent trajectory. Okay, for me, that's, I mean, first time I saw that, that was really shocking. Okay, of course, I mean, did I show something? Yeah, here, people found similar, this kind of spiral waves or rotational waves based on ECOG recordings. Okay, for sleep spindles as well, but Terry Snow group and the Salk Institute. Okay, they also found that those spirals, okay, very consistent as well. Okay, but for the first time, we linked those spirals to behavior, and also this long-term consistency is a good index of aging. Okay, as we are getting older, the consistency would decrease. Okay.
So it means the long-term consistency increases, and then the patient perform better?
Yes, exactly.
After
after that, yes. So, yes. Yes, after. So far, we only have 9 subjects, yes. Yes, we actually look at other indices or quantities, as you mentioned, the durations of those spirals and the amplitudes of those spirals. We couldn't find significant correlations, okay, but only the trajectory, okay, for the reason I've just mentioned, because you need to have this kind of more stable dynamics to consolidate, to have more memory consolidation. And also you can have these sort of bigger waves, okay, sweeping through your brain, because this is important in terms of that, because you have this rotational direction, okay, motion, right? So it can integrate information you acquired during daytime, okay, for different modalities. Let's auditory information, visual information, or some other information across different cortical areas. And that is because of the rotation, okay, so these activity patterns could integrate information so that information you acquired during daytime could be further processed. Okay, that's why it's related to memory performance, related to memory consolidation, I think. Okay, but that's just one assumption, right? You can come up with another assumption, maybe for your project for this summer school. But anyway, so that's related to this performance and the behavior.
Excuse me? I have a question.
Yeah.
About the previous plot. How is the long-term consistency measured? Like, what's 0.6 means?
Oh, so this is a long-term consistent because you have one trajectory, right? And for one trial or for one subject, and then we introduce the cross correlations. And first you get a map, okay, average, the trial average, the subject average map, and then you look at the correlation between that map and the individual subjects, and then you can come up. Because if individuals are consistent with the average map, okay, we will say, oh, it's very consistent. Okay, so based on that, we come up with consistency map. And this is just pure theoretical predictions or one of my assumptions. So I think those wave patterns like spirals or source or sink or maybe many other patterns, they may allow or enable the brain to a non-conventional computing. Okay, so the idea is that so the collisions or interactions of those patterns may do a type of distributed dynamical computation. Okay, you can think about if you have a wave traveling from here to there, and then you have another wave traveling from here to there, they interact, right? Because of these interactions, information can be processed and can be integrated, and you have many waves working together, performing distributed dynamical computation. Okay, so it means that, I mean, if this assumption is right, it means that the propagation of wave can be used for communicating information. Okay, and when they interact, either interference or this collision, when they interact with each other, information could be processed. Okay, so this information processing is very transient phenomena. It's not like a attractor or something like that. Okay, purely based on these dynamical interactions of different types of waves. So this idea actually is very analogous to classical non-conventional computing paradigm proposed by Fredkin and Pulin. So Fredkin actually, he was a theoretical neuroscientist. He died actually 2 years ago. So he published a paper in 1982. So the idea of this proposal is to link physics with computation. Okay, so this is a collision of two physical balls. You have one ball coming from here, you have another ball coming from there. These two balls would collide. And you know, as you learned in high school or first year physics, so this collision, okay, often we assume this is elastic collision. And so the idea of this is that they're trying to link this elastic collision with computation. Okay, with computation, as you can see, so this when both balls, this comes from here, this comes from here, but both of them, they are here, you say you'll get a P and a Q. So this is logical computation, logical gate. It means A and a B. One is here and another is not here. It means that you detect one ball here. So this is not that P is not there, Q is not there. Okay, not and logical operation, and so on. So because you see these logical operations are complete, it means that by setting up elastic collisions of those balls, you can do universal computation. Universal computation. Because based on these logical operations, you can set up any computation. Okay, but this computation is different from like computation underlying Turing machine, because here you exploited like elastic collisions of the physical balls for doing computation. So this paper actually, I mean, because at that time, I mean, they were at the California Institute of Technology, right, Caltech, and also Feynman was there as well, Richard Feynman. And motivated by this paper, Richard Feynman wrote that very revolutionary paper with the title of How Does Physics Compute? So that paper actually initialized the field, the whole field of quantum computation. Because for quantum computation, so you wanted to link quantum physics with computation for non-conventional computation, right? And also around the year, John Hopefield, I mean, people talk about Hopefield neural networks, right? Hopefield published his landmark paper about attractor neural network, okay? Because at that point, those guys, okay, they were working in the same building, okay, they discuss, at that point they discuss how the universe does computation. And also relate to that, Wolfram, okay, Stephen Wolfram, I mean, he has done a lot of work on computation of cellular automata. Based on that body of work, he published a book with the title of New Kind of Science. Okay, so the idea is that the whole universe may do some sort of computations. But anyway, so that's just sort of like practical predictions. Okay, and so that's for the methods we've developed, and based on these methods, we made some new discoveries. Okay, such as source or sync patterns or sending patterns. For the first time, actually, we found in the real brain. There will be a following up paper, I think, from a group from Columbia University. They have a paper which is now, I think, on the press, okay, in press with Nature Neuroscience. Okay, they found something for source or sync, all those kind of patterns. And then, I mean, as I said, okay, I'm a theoretician. Okay, I wanted to, of course, wanted to have a little deeper understanding about how those patterns can emerge from the real neural circuits. Okay, so of course I will talk more about that topic from a more advanced lecture. For now, okay, I'm going to introduce model published in 2015. So this computation model is physically realistic. So this is, you see, we have capacitance. Okay, so this is reluctance. Okay, this is reverse potential, okay, for leaky reverse potential, and this is for excitatory neurons, for inhibitory neurons, okay. As in real neural circuits, okay, we have excitatory neurons, we have inhibitory neurons, okay. The number of excitatory neurons is 4 times that of inhibitory neurons, okay, 4 to 1, as in realistic network. And also different, like random neural networks, okay, for this realistic network, we arrange all neurons on this 2D space, okay? And also, as you can see, so this is the synaptic conductance, okay, to individual neurons, lambda here, lambda is E, it is for excitatory neurons, while lambda R, that's for inhibitory neurons. So this is coupling strength. For this realistic network, I mean, the coupling strength is function of distance. It means that when neurons, when they are further away from each other, they would have less coupling strength. Okay, distance-dependent coupling is found in the real brain. And also we consider this realistic conductance synaptic dynamics as indicated here, because people use this to fit the postsynaptic current, okay, conductance, okay. And for, you see, we just set up this computational model. And so this is external input. For our case, we put this as constant external input. It means that we want to see whether this sort of complexity of patterns can emerge from a deterministic system rather than being the byproduct of some noisy factors, right? So we set it up in this way, and also we made sure that the network is balanced in the sense that synaptic current, like excitatory current, inhibitory current, so have this ratio between excitatory current and inhibitory current, the ratio is close to 1. It means that excitatory synaptic current and inhibitory current, okay, they somehow are balanced as found in the real neural circuits. And then, I mean, you can set up your numerical model, okay, and then you can do simulations. So immediately when we set this up, and then we can do simulations, right? And then you see in this balanced stage, it's a stage when the synaptic inputs to individual neurons are balanced, you'll see this sort of emergent, this kind of localized coherent structures or wave patterns, okay? And so I didn't show a movie here, but you will see, so this pattern actually rotates, okay, in this way, this one may rotate in that way, and so on. And also you have this kind of a pattern, right? This pattern would spread out from this region, okay? Bigger and bigger, have this kind of expanding action. So this may be a source pattern, as I said. Anyway, based on this realistic model in balanced state, okay, for deterministic model, you'll see this sort of emergent coherent structures. And when excitation is stronger than inhibition, you will see these beautiful stable spiral waves, okay, something like we found for spiral waves of sleep spindles. Okay, they are very stable, okay, rotate in that kind of very regular way. So it means that, so maybe stable spirals in the sleeping brain could be related to this kind of modulation for balance of excitation and inhibition. So one thing we can learn from this modeling work, so balanced excitation and the inhibition as found in the real brain is important neural mechanism for the emergence of those complex wave patterns. But things that actually, just based on this numerical model, right, we can integrate different ideas. Okay, that's the strength of modeling work. We can integrate different ideas about the mechanism of irregularity or variability of spikes. As I said, okay, neurons, they fire very irregularly, okay, variably. And there are two competing ideas proposed once by Sompolinsky and his collaborator, which is this balanced neural network. But for their balanced neural network is for this kind of like randomly coupled neural networks, right? And another idea, as found by Zander and in his group, and also another group, they found that so for this idea, it proposed, you are in balanced state, neurons are not correlated, okay, in asynchronous state, okay? But for this idea, they actually found that the inputs to individual neurons are very correlated, because in theory, so these inputs to individual neurons can be formulated as Gaussian process, right? But when you look at the raw data, so this is based on data, you found that actually synaptic inputs to individual neurons would have this kind of bumpy behavior. Sometimes you have a lot of input, sometimes less, sometimes a lot, sometimes less. Okay, so it means that for the real brain, if we want to explain this irregularity, okay, so we get somehow balance this kind of neurophysiological mechanism to correlated synaptic inputs. Okay, so it's kind of seemingly contradicting ideas, but actually that can be integrated by our model. That's why our paper actually was selected as a cover article of Journal of Neuroscience. Okay, so because things that also you have these coherent structures emerging. You see, when you have pattern here, so neurons along that local area, okay, they will fire a lot. Okay, they would fire a lot. When the pattern moves away, they fire less. So you have these kind of fluctuations of neuron spikes, and then. So this only happens when the network in balanced state. So that's related to balanced idea. But during this process, okay, as I said, when the pattern is here, you have a lot of synaptic inputs to those neurons, you will get this kind of bumpy intermittent synaptic inputs, okay, as found by others like Zander's neurophysiological recordings. Okay, in this sense, our numerical model can integrate the idea of synchronized inputs and the idea for balanced excitation inhibition for explaining the irregularity of neural spikes. But more important than as I said, okay, beyond irregularity, okay, and the circuit level, we have these emergent coherent structures. Okay, so in this sense, you see that's a strength of modeling work, right? We can integrate neural dynamics across different levels and come up with a neurophysiologically realistic explanation. Okay? Yeah. 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 I think that's a fair question, okay? But it does have function. And I'll demonstrate it based on the data, right? And also in my second lecture, okay, I will show you those patterns actually can be used to explain many cognitive functions. Okay, we've been collaborating with Pascal Fries, okay, the guy who made this assumption about gamma synchronization, which can be used for communicating information. So we wanted to test this kind of theoretical proposal as well. But I think your point is fair point, okay? Or I mean, yeah, that's a fair question, yes. And then you see we can calculate the final factor, okay, which is variance, okay, of spark count within a time window divided by mean. So this is our prediction. You will see this kind of increase, okay, of final factor, which is about the variability of neural fire rate, okay, the increase. But for the conventional theory based on something proposed in this paper, final factor is flat. Because in that theoretical model, neurons would fire in a Poisson manner, right? For Poisson spikes, variance is the same as mean. And then, so, okay, anyway, so this is some modeling work, and we can link different experimental phenomena together. And finally, because this is an introductory lecture, okay, I'll quickly go through. Maybe you will cover this or not? No, okay. But anyway, so this is based like neural fields, okay, for studying pattern formation in neural circuits. So this is one of the first papers on this topic by Shuichi Amari. So in this idea, I mean, which is different from the spark neural circuit model, I'll just demonstrate to you. For this one, we assume homogeneous neural field, okay? All neurons, they behave similarly. For this case, there's no separation between excitatory inhibitory neurons. So homogeneous field. And the population activity, okay, is this average memory potential as UXT. And this is function of time because it changes, okay? And this is this term, and also this is the synaptic coupling strength. So this is activation function, and for this paper, classical paper, so this is assumed to be a Heaviside function. If the input is greater than 0, I mean this is 1. If the input is smaller than 0, it's 0, okay? So this is a simple Heaviside activation function. H is a constant homogeneous external input. Of course, you can add time varying external inputs to your neural field model. So this model is called the neural field model, okay? So you may read some papers, people talking about spark neural circuit models. Okay, I'll just demonstrate to you, and the neural field model, and also neural mass model, okay? So for the homogeneous field, okay, and we assume, okay, the coupling strength, which is W is only dependent on the distance between two neurons or between two populations. And then, and also Amari assumed that this coupling strength is this kind of masking height function. For nearby neurons, okay, we have this kind of excitatory interactions, okay? When neurons, they are a little bit away from each other, we have inhibitory interactions, this sort of masking height, this kind of shape of synaptic coupling strength. And then, as usual, okay, and like if you have a dynamical system like this, first we want to look at the equilibrium state of this system. So to do that, okay, we just assume this term is 0, okay? So while this term is 0, what it means that U, which is potential is not going to change because this derivative with respect to time. But this is not change, it means that you have fixed point, stable state, okay? So it means that the whole term is 0, and then based on this, okay, we can come up, because the whole term is 0, you arrange this to left side, and then you can come up with this equation very easily. And for this one, I mean, actually, I mean, we are interested in this sort of local bump, okay, local bump, this sort of pattern, okay, again, pattern. As I said, this is first paper on pattern formation. We want to see how this stationary pattern can be formulated in neural field model. And then so we have this equilibrium state, and because of this assumption about Heaviside function, and very easily we can get this U equals to this term. So this is a local excitation region, okay, localized excitational region, and we only within that region, okay, so we have this term because this Heaviside function, okay. And then based on this, okay, as I said, okay, and because the interaction is only dependent on the distance between two neurons, if we have this local activated region from A1 to A2, and we can shift it from 0 to A, okay, which is because they are equivalent translational invariant. And then based on this, okay, here, I mean, we can do some mathematical analysis. I will skip this, okay, but you can look at nodes. Okay, I mean, I use this for honors students and the University of Sydney. And so, but anyways, you can look at the duration up down there. Okay, I'll stay here until Saturday. If you can't understand this, I mean, you can ask me questions. Okay, but anyway, very basic dynamical systems analysis, and based on this, we can figure out. So this is a standard Taylor expression. Oh, sorry. This is standard Taylor expression to look at perturbations in space and time. And based on these perturbations, we can figure out the stability of the equilibrium state. And then we, so some, I mean, based on this, we can come up with stable and unstable conditions of that localized bump pattern, okay? And to tell probably how the patterns can emerge from this near field model, okay? And this is a summary of that theoretical result, okay? So as I said, you can look at my notes, particularly from dynamical systems theory aspect. I think I'm just on time. Okay, here, a summary. So I think the first idea you should get is that, okay, the brain exhibits very complex and rich spatial temporal dynamics beyond synchronization, beyond cross correlations, okay? And also to detect those spatial temporal dynamics, we developed a methodological framework, okay? So you can check our methods paper, and also we applied the methods to large-scale recordings from different modalities, local field potentials, and high-density EEG for different states, anesthesia states, wake state, working brain, and also sleeping brain. And we made new discoveries for spirals, and also we found that the interactions for those spirals are very fundamental in the sense that they can be used for organizing this kind of large-scale brain dynamics, okay? And also we found that they are related to functions. And finally, briefly about spark neural circuit model we've developed, okay, based on that model. Although external inputs are deterministic, we can see these sort of emergent complex wave patterns. So those wave patterns, their dynamics can unify two different ideas proposed for understanding the irregularity of neural spikes. And also we briefly went through the classical pattern formation paper done by Professor Shuichi Amaru. Thank you.
So you were talking about the singularities of those spirals on the boundaries. What does it exactly mean when they're on the boundaries?
Yeah, I think yes. As I said, I mean, as I said, we've been developing model trying to understand why they would emerge from the boundaries between different functional areas. But from functional aspect, so you think about it, because I think this is very strategic location, right? Because if spirals are around boundaries, right? Because of the rotational motion, right? Rotational motion. So this is extra degree of freedom as compared with the planar wave, right? So this rotation can organize the activity to flow out from one functional region into another functional region. Otherwise, I mean, what's the mechanism for coordinating different functional networks as found by neural imaging field? So what's the mechanism, I'll ask you. So what would be the mechanism for coordinating the flow of activity or communications between different functional networks? So based on your assumption, let's say. Yeah, try to be creative and come up with some assumptions. We can discuss that later.
Yeah,
doesn't matter, yeah, doesn't matter, yeah.
Let's keep the question short because we have...
Yeah.
What about other points like unstable fixed point or settle point? Would there be a pattern like that?
Yeah, we found in the MT area of marmoset. Yeah. Thank you.
I have a short question. Sorry.
Yeah.
So, do you consider long-range projections in the data analysis? Like, so will the long-range projections between brain regions?
You mean connectivities?
Yes.
I mean, for this is let's say activity data, right? I mean, we can't do that sort of analysis. But of course, long-range connections could be important in terms of the emergence of those patterns, okay? Because most of the connections are local, distance dependent, but we do have this kind of long-range connections. They could be important for the emergence of those patterns, yes.
Thank you.
That's a very good question.
Professor, how many neurons think you need to do this kind of analysis?
Oh, I mean, for example, for local field potential recordings, we have 10 by 10, so 100 electrodes. And for fMRI data, you know, that's a lot of sensors, right? Voxels. And for high density EEG, we have 256 electrodes. But you get to have enough recording size to cover spatial structure. Okay.
Do you think in some